For this lab, we'll be going into the nervous system as well as our senses. So let's just jump right into it and get through this lab together. If you remember from the skeletal system, you split the skeleton into two divisions, the axial skeleton and the appendicular skeleton. We can do the same thing with the nervous system as well, although the naming is going to change a bit. Remember with the skeletal system, the axial skeleton composed of the skull and the vertebral column and ribs. Everything was along the midline, and the appendicular skeleton was everything from the shoulders and arms, hips, and legs. We see a similar division in the nervous system, only instead we're calling it the central nervous system and the peripheral nervous system. The central nervous system is composed of the brain and spinal cord, and the peripheral nervous system is all of the nerves that extend out of that. So let's dive into the central nervous system first and look at how the brain functions and controls overall how we interact with our environment. The brain's main function can be broken down into three parts: sensory input, interneuron processing, and motor output. Sensory input is the act of bringing information in, either from our own body or from the environment around us. Interneuron processing is taking that information and making sense out of it. What does this information mean, and what do I want to do about it? And then lastly, motor output is where we send a signal out to perform an action in response to the information that we received. So if we were to put this together in a scenario, let's say I'm at the park with a friend and I'm throwing a football around with them. The sensory input that I would have would be visual. I would see a football coming towards me. Possibly auditory. My friend may yell "catch" to me, as well as information like the wind against my skin and the grass under my feet. All the information around me would be coming in. We then have to process that information and make sense of it. I need to bring that visual information in and understand what I'm looking at, recognizing that there is a football there, recognizing that the football is coming closer to me, hearing what my friend says and understanding the word "catch," knowing what to do in response to that, and then deciding what to do with this information. I'm in a park, I got that information from the environment around me, I see a football coming towards me, and I hear the word "catch." I can then send a signal down my motor neurons to my arms to tell my arms to reach up and catch the football. In this case, my muscles in my arms would be the effector, the ones performing the action. So just to recap, sensory input is where we bring information in from either our own body or the environment around us. We then use interneurons to process that information and make sense of that, and also decide what to do with that information. And then motor output, we're sending a signal to cause an action in response to the information we got. Now let's look at some of the major structures of the brain. Now I don't have everything listed here. We're going to keep it short and sweet to about 4 or 5 main structures or main divisions. And then we'll talk about some of those parts in the lab. You're going to go into more detail in your lecture, so just keep that in mind when you study. We're going to keep it relatively low-key for lab, but you'll get into more detail in lecture. So we can break it down into 4 to 5 groups: the ventricles, the cerebrum, the diencephalon, and the cerebellum. One that I don't have written down on here is the brainstem, which you should also be familiar with. Let's go into each of these. The ventricles, which you can't really see here, are open spaces that are filled with cerebrospinal fluid. The cerebrum is probably what you think of when you think of a brain, the wrinkled bits. We can divide that into lobes or sections, and this is where our conscious thought and our processing of all the information we get is going to happen. The diencephalon is seen here in gold. This is a fluid-filled cavity that has the thalamus and hypothalamus as part of it. We'll talk about each of those. And then back here we have the cerebellum. The cerebellum is going to help kind of bring together all the information that we need to modulate a response, to coordinate something. So damage to a cerebellum would lead someone to have trouble coordinating multiple movements at once. The one I don't have written here is also the brain stem, so let's talk about that. Made up of the midbrain, the pons, and the medulla. And this is the very primitive part. So things like making sure that you breathe, making sure that your heart beats are going to be controlled by the brain stem. Now, we're not going to get into all of those 5 groups that I just showed you. We're going to only get into a couple, mainly the cerebrum and the diencephalon. So let's get into the cerebrum first. Like I said, we divide the cerebrum into lobes or different sections. And we have them color-coded here, with the frontal lobe in blue, the parietal lobe in pink, the occipital lobe in purple, and the temporal lobe in red. Now, keep in mind, we're going to simplify it down when we talk about what each lobe does. Do recognize that the brain is very complex in how it handles information. And so, it's not going to be as cut and dry as what we're going to describe it in reality. But do recognize that these are at least some of the basic tasks that these lobes handle for the most part. The frontal lobe in blue is going to be where our higher thinking happens and our conscious thought. It's also where we have voluntary muscle control and have our control for the different motor functions we perform. The parietal lobe in pink, just kind of sitting behind it, is going to deal with somatosensory information, meaning information that comes from your skin and some muscle, as well as taste. The occipital lobe in purple will handle visual information, and the temporal lobe in red will handle hearing, as well as integrate the different senses with memories that we have. The diencephalon is mainly composed of 2 pieces, the thalamus and the hypothalamus. The thalamus is going to take sensory information in before it gets to the cerebrum, and it's going to serve as a gatekeeper. So we can portion off the sensory information to the right sections of the brain so that we don't send visual information to the frontal lobe if it's not supposed to go there. The hypothalamus is somewhat of a primitive part of our brain. It's going to control basic things like appetite, maintaining your body temperature and water balance, as well as handling things like pleasure, reproductive behavior, hostility, and pain. The hypothalamus also connects with the pituitary gland. We're not going to get into that here, but we'll hear more about the hypothalamus and the pituitary when we get to the endocrine system. Now, the brain, given what it does and all the important things that it can handle, needs protection. We get protection from things like the skull, as well as the cerebrospinal fluid. We also have one we haven't mentioned yet, the meninges. We see the meninges here. It almost looks like plastic wrapped around the brain here. And it's composed of 3 main layers, the pia mater, the arachnoid mater, and the dura mater. These meninges are going to kind of encapsulate the brain and spinal cord and give us some extra protection on top of what the skull and cerebrospinal fluid provide. Speaking of spinal cord, let's go into the second part of our central nervous system, which will connect us to our peripheral nervous system. The spinal cord is protected by the vertebral column. Just like the brain is protected by the skull, the spinal cord is protected by the bony vertebrae, forming a cage around it. And we find both in the brain and spinal cord that we have 2 types of brain and spinal cord matter, gray matter and white matter. Gray matter is involved in the processing, taking that information in, putting it together, and making sense of it. And so we contain the nerve fibers, those interneurons involved in step 2, right, the interneuron processing stage, as well as the cell bodies of the motor neurons. The white matter is involved in transmission, sending those signals up and down the nerves. They're long fibers and they have the insulating myelin sheaths that give it the white coloring it has. So let's look at a transverse section of the spinal cord and the nerves that are exiting out of it. So here we have the spinal cord, and this butterfly shape you see in the middle there is the gray matter, whereas the beige around it is the white matter. Extending off of it from either side are going to be those spinal nerves. So information will be carried to and from the spinal nerves to the spinal cord, processed in the gray matter, and then any sort of effect that we want to happen can be sent down through a motor nerve. Now, if you're looking at this picture and getting nervous because of all the things that are labeled, don't worry just yet. You're not going to need to be able to label all of these components. You really should just be focused on knowing the gray matter versus the white matter and being able to identify that on a picture like this, as well as the spinal cord and the nerves. Let's look at a real picture of a spinal cord, as well as the nerves exiting from it here. So we've cut away all the other tissue and have fully exposed that spinal cord. Which leads us into the spinal reflex. Now, we've really just talked about the brain doing a lot of this processing, but it's not the only part of your nervous system that can process information. In some cases, going all the way to the brain is just too slow, even given how fast that reaction is. Sometimes we need a reaction to happen even faster. Let's say in the case of you accidentally touching a hot stove. In that case, we have the spinal cord, which allows us to create a reflex arc or a spinal reflex, where skeletal muscles will react that require no connection to the brain whatsoever. Instead, we use that gray matter found in the spinal cord. So the pathway, instead of going all the way to the brain, will go from a sensory receptor to a sensory neuron, to the spinal cord, then to a motor neuron, and then finally the effector, which in this case would be a muscle. So let's create a scenario to kind of describe this process in action. If I touch a hot stove, there's damage that's occurring. My skin is getting burned. We need to be able to pull that hand off of that stove as fast as humanly possible. And that means we're not going to be able to go to the brain because it's going to be a little too slow. We have a faster way to move that hand off of the stove. So in my hand, as I'm touching the hot stove, I have sensory receptors detecting the heat and detecting pain. We then send that information along a sensory neuron to the spinal cord. The spinal cord realizes that there's damage occurring to my body and sends a signal down a motor neuron to my arm to cause my muscles to pull my hand away from the stove. This is happening near instantaneously. By the time I've pulled my hand off, I haven't even realized that my hand hurts yet. Moving forward, we go to the peripheral nervous system. This is all the nerves that connect to both the brain and the spinal cord, and this is what makes it possible for us to really interact with and understand what's happening around us. And there are different types of receptors that help us bring that information in. This is the starting point when it comes to communicating information to the central nervous system. And we have several different types of receptors that bring in different types of information. We're not going to get into all of them. You'll probably go into more in your lecture. So we'll go into the short list really quick. Mechanoreceptors detect things like physical changes, things like pressure and touch. Chemoreceptors detect chemicals, so different molecules will trigger chemoreceptors. Photoreceptors detect light, and a special type of one that's called a proprioreceptor will help you maintain things like balance and posture. So let's get into a few of the senses. We're going to talk about taste, smell, sight, and hearing. Taste and smell I have together here because they're very closely linked together. Both your sense of smell and taste are detected by chemoreceptors. We're detecting the chemicals in the food we eat and the air we breathe. So taste receptors are found in the taste buds, and the olfactory receptors are found in the roof of the nasal cavity, handling smell. And we see both of them here. So from the food we eat, right, molecules will trigger the taste receptors, which will send then a signal down through a nerve to the brain. And then up here, olfactory receptors will detect the molecules in the air we've breathed in and take that information down from the olfactory bulb and olfactory tract down to the brain again. It's important to note that these two are very closely related. Your sense of taste is pretty basic. Salty, sweet, savory, bitter, and sour. We don't have a lot of nuance when it comes to taste. Your ability to tell an orange from a lemon doesn't come down to your taste receptors, it comes down to your olfactory receptors. So your olfactory receptors help refine your sense of taste so that you get those nuances. You can tell the difference between things that are in the same categories. Otherwise, if you tasted a candy that was lemon and another one that was cherry, since they're both sweet candy, they would taste exactly the same without your sense of smell. About 80 to 90% of your sense of taste comes from your sense of smell. And this is why when you get sick and you have a lot of congestion, food can sometimes be really, really bland. It's because the majority of your sense of taste is not coming from your tongue at all. It's coming from your nose. Let's now get into vision. We're not going to talk a ton about the physiology behind vision, but we're going to at least get into the basic anatomy. And I have it broken down based on the kind of function or part of the eye that it is. So let's start with the layers first. We have three main layers to the eyeball. This outer white sclera, which provides some protection and structure. The middle layer called the choroid, which has blood vessels and pigments. And then the very inner layer shown here in gold, which is the retina, which will have the photoreceptors embedded in it. When light comes in, it's mainly focused and refracted by the lens. But since we can look at things both near and far, we have to be able to change this lens's shape so that when it refracts that light, it always focuses it back on one single point called the fovea centralis. In order to change the lens's shape, we have the ciliary bodies and suspensory ligaments that will help move this lens either by flattening it out or rounding it out so that we always focus light on the same point in the back of the eye. We can also break the eye down into compartments. The anterior compartment up in the front, filled with the aqueous humor, and the posterior compartment filled with the vitreous humor. When light comes in the eye, we have to go through several structures in order to do so, and we have the ability to control how much light comes in. The cornea is an extension of the sclera. It's been modified so that it is clear, so that light can pass through. And then we have the pupil and the iris here. The pupil is the area where light actually comes in and passes through, and the iris will control the size of the pupil. So the iris is the part of your eye that has color. Maybe you have beautiful chocolate browns or cute baby blues. But no matter what color your eyes are, your iris does the same thing. It's going to help control the size of the pupil. So if I were to shine a flashlight in your eyes, your pupil would get very small because your iris would make it smaller to make sure that too much light does not enter the eye. Opposite of that, if you're in a very dark room, your iris will make your pupil much larger because there's not a lot of light available and we need as much to come in as possible in order to see. After we pass through there, we can pass through the lens, which will then again refract and focus that light in the back of the eye. When it comes to bringing in that visual information and sending it to the brain, we have the optic nerve, fovea centralis, and rods and cones embedded in the retina. The optic nerve here will connect to the brain so that we can pass visual information on. The fovea centralis is this portion here where we have a densely packed area of cones, of photoreceptors. The photoreceptors, the rods and cones, will detect light, rods detecting the level of light, and cones helping detect color. It's important to note that where the optic nerve connects with the human eyeball, there are no rods or cones. This forms what we call a blind spot, an area where there's no visual information coming in. There are tests that you can do with just a piece of paper and a hand covering one of your eyes so that you can find your own blind spot. I recommend trying it out. It's a pretty fun little thing to do. So now we've gone all over every structure that we are going to really worry about in lab for the eye. Let's get into hearing and the ear. We can break it down into three sections: the outer ear, the middle ear, and the inner ear. The outer ear is what I like to call all the parts you can touch with your fingers. The pinna, which is the part that you think of when you think of an ear, right? The part that is on the outside. And the ear canal form the outer ear. The ear canal is the area where you could stick a Q-tip in, although you really should not do that. As we go deeper in, we get to the tympanum, or the tympanic membrane, also known as your eardrum. The eardrum is the first structure of the middle ear. followed by the ossicles. The ossicles are a collection of 3 little modified bones that will help amplify the sound coming from the eardrum. Individually, these ossicles are known as the malleus, the incus, and the stapes. You should know all 3 of them. So when sound comes in, the pinna directs sound in towards the ear canal. The ear canal continues to direct sound waves towards the eardrum. The eardrum will vibrate, which will then cause the malleus, the incus, and the stapes to move in response, and they'll amplify that signal. Our stapes will then hit the oval window, and we then get into the inner ear. When we do that, we will work with the cochlea. Inside the cochlea, we have little hair cells that will detect physical changes, physical movement of little hairs, little cilia on those cells. And it's the movement of those little cilia and little hairs that will help us determine if something is loud or quiet, and if it's a high pitch or low pitch. So the cochlea is our organ for hearing. But it's not the only component found in the inner ear. We also have components for balance, namely the semicircular canals and the vestibule. Those will help determine your location, whether you're looking up or down or turning to the sides. One of the last things we have is the eustachian tube, also known as the auditory tube. So if you've ever gone flying or gone to a higher elevation, you may have noticed that your ears feel like they're popping. When it comes to air pressure, if you change an elevation, like going in an airplane, you change the pressure that's on the inside versus the outside of your eardrum. Now, if you had no way to protect yourself against this change in pressure, your eardrum would get blown out. However, your auditory tube, which connects to your throat, will help regulate that pressure so that it'll equalize on both sides of your tympanic membrane. So altogether, you should know the structures of the outer, middle, and inner ears. You should be able to tell me which structures are involved in hearing and which ones are involved in balance. And you should be able to tell me the nerves that are involved, which I just will get into right now. The vestibular nerve will work with the balance organs, and the cochlear nerve will work with the cochlea. Definitely take some time to get to know the eye and ear. You will be expected to be able to identify some of these structures from pictures, so you don't want to gloss over them too much.